We'll now look to Dr. Natasha Chang, who joined McGill University as an assistant professor in the Department of Biochemistry in 2019. Research in Dr. Chang's laboratory focuses on understanding the molecular signaling mechanisms that regulate muscle stem cell function and how these pathways are altered in the context of muscle degenerative disease, and muscle cancer. The ultimate goal for Dr. Chang's research team is to identify molecular targets to improve endogenous stem cell regenerative capacity, as well as strategies to improve stem cell transplantation therapy. Tonight, Dr. Chang will give us an update on her research into inducing stress granule formation in muscle stem cells to treat DMD. Welcome, Dr. Chang. Thank you so much for joining us.
Thank you so much. I'm going to share my screen. Does that look okay on your end?
It looks great. Thank you.
Okay, perfect. So thank you again for giving me this opportunity to present an update on our project. So our approach to Duchenne muscular dystrophy has really been focused on looking at the muscle stem cell. So these are highly specialized cells that reside in the muscle tissue, and they are also known as satellite cells. So that's the cell that I'm showing here. So they sit on top of the muscle fiber, and they're responsible for sustaining the regenerative capacity of our muscles throughout life. And the research in my lab has been focused on understanding these cells in a healthy muscle as well as in a degenerative context. And we know from a lot of work in the field that there's increasing evidence demonstrating that the muscle stem cells in DMD are not functioning the way normal healthy muscle stem cells do. And so what I'm showing here, these are just some examples of the impaired molecular and cellular properties that have been observed in muscle stem cells in Duchenne muscular dystrophy. And so our lab has really started to make some progress in understanding at a single cell molecular level how DMD muscle stem cells are different from muscle stem cells in healthy tissue. And so we've performed what's known as a really highly sensitive experiment called single cell RNA sequencing. And what this told us was that muscle stem cells in DMD exhibit increased cell death, that they are impaired in their ability to enter the cell cycle, and they have impaired cell survival pathways. And most importantly, they have a defect in their ability to make muscle. And so the way that we think about DMD is that this is both a disease of the muscle tissue, where there's a loss of muscle integrity and chronic degeneration. But at the same time, it's also a disease of the muscle stem cell, and their dysfunction contributes to the inability of the muscle to undergo efficient repair. And so our strategy is really based on how can we restore muscle stem cell function to make better muscle. So the rationale for our project came from previous work done in the field of muscle wasting in cancer-induced cachexia, which is a severe muscle wasting disorder, where treatment of both cells and mouse models of cachexia with a drug that induced formation of these cellular entities called stress granules resulted in improved differentiation and improved muscle function. And so what we were asking was if these compounds would also work in DMD cells and animal models. And so we treated DMD muscle progenitor cells with a drug that induces stress granule formation. And we asked if they can differentiate better in the presence of this drug. And the answer is yes, we see improved fusion of these muscle progenitors into muscle fibers, which is a readout for differentiation. We've also now confirmed that treatment with this drug does not impact cell survival or the ability of DMD muscle progenitors to proliferate. So here we are validating that the treatment is not toxic to the cells. And we've been asking if this compound can improve muscle regeneration in mouse models of DMD. So we've injected DMD mice with the compound, and we found that there is this slight increase in the muscle weight upon treatment compared to our control mice. And it was important for us to assess the number of muscle stem cells following treatment because we don't want to deplete the stem cell pool by pushing the cells to differentiate. And so far, we don't see any change in stem cell number following treatment. And we also checked for how other cell types in the muscle would respond to the treatment. So we looked at cells involved in inflammation. And what we see is an increase in the number of these pro-regenerative macrophages. These are cells that actually help the regeneration process. So this is actually quite an exciting finding for us, and we're following up on this. So as we continue to work on this project, we're focused on studying the molecular mechanism of how this compound works. What is it actually doing in the cell to promote differentiation? And we're also continuing to add more mice to our studies to demonstrate the effect of the compound. And we'll be testing this also in DMD cells from patients to confirm that what we're seeing in mice will also work in human cells. And so that is the research update I have for you. I would like to thank, of course, my collaborators for their contributions for this work, and my whole lab. We really function as a team. And I'd like to thank everyone in the Defeat Duchenne Canada community for supporting our work. Thank you very much.
Thank you. And thank you for such a great update. I appreciate it.